Hello, and welcome to the BYU Family History Library webinar series. I'm Marin, and I'll be your host for this webinar. Please make sure that your microphones and web cameras are disabled during the presentation to provide a smooth webinar. Our upcoming webinars are on August the 15th, so that'll be Thursday at 3 p.m. Mountain Time. We'll be having a webinar with Bob Taylor. He's going to update us on all the new things that are on the Family History Guide. Then after that, we'll have a webinar with Catherine Grant. She'll be going over the helper resources for the Discover, Gather, Connect. And then to end the month on the 29th, we'll have a webinar with Sarah Stoddard. She'll be giving a follow-up on DNA. If you'd like to access a previous webinar, please visit our webinar index on our website or search our YouTube channel. All of our webinars are recorded and uploaded the following Monday for your convenience. We also post links to the recordings as well as updates on our Facebook and Twitter accounts. During the webinar, if you have any technical difficulties, please use the chat box and I can address those concerns. You're welcome to use the chat box during the webinar for comments and insights. However, all questions will be addressed at the end of the presentation. For today's webinar, we're pleased to hear from James Tanner, who will be giving a presentation on the history of the development of genealogical DNA. James Tanner has over 32 years experience in genealogical research and is an avid blogger of Genealogy Star and Rejoice and Be Exceedingly Glad. He has presented at expos and conferences around the US and Canada. He served for 10 years as a missionary at the Mesa, Arizona Family Search Library and is currently serving at the BYU Family History Library. James has 7 children, 34 grandchildren, and 1 great-grandchild. And if James is ready, then I can get that presentation set up.
Okay. Howdy. This is James Tanner. James Tanner here from the BYU Family History Library on Brigham Young University campus in Provo, Utah. We're going to be talking today about the history of the development of genealogical DNA testing. Now, this is not a technical presentation about how DNA is used to find your ancestors or identify your relationships in your family and all that. This is a background. This will give you some perspective about how relatively new DNA is as a subject and as a testing. So this will talk about the history that goes back only a very relatively short time as history goes back. So starting out, one of the things that we need to know as we get into the use of DNA for discovering our ancestors and our relatives is that the impact of genealogical DNA testing is going to turn out to be fairly disruptive and probably deeply disturbing to a lot of people. Not just because they are surprised by what their ethnicity might be or whatever, but we are really coming up with some interesting situations. And quite regularly now, I am talking to people. In one instance, a person that I know ended up finding out she had 3 new first cousins she didn't know about. And the situation there was quite, let's just say, interesting and complicated. In another instance that's been quite recent, I've been helping someone who found out that they were switched at birth and that the family they'd been doing research on for 35 or plus years were not even relatives. This is the kind of thing that is happening quite regularly. It's going to restructure a lot of families. A lot of the information that we're getting is really quite new and not well known in families simply because of cultural reasons that these particular problems have not been well represented or well made available to family members. Another thing we need to know about DNA testing is that the beneficial effects of a DNA test attenuate with each generation going back in time. In other words, the further back in time you go, the more difficult it is to be specific about the DNA testing. And in this case, and something that I will probably repeat, is that DNA testing does not replace your paper genealogy, meaning that the research you do as a genealogist in creating a family tree, which is based on records and sources, is not being replaced by DNA. It is being supplemented by DNA, and DNA has become a genealogical tool, but it is not necessarily the end product of everything that we want to know. And there's Another part of this that needs to be reviewed in a little more detail is that there's no specifically clear definition of ethnicity. Everyone who does a DNA test through one of the large genealogically related DNA companies today gets an ethnicity estimate. And the question is, of course, what is ethnicity and what does it mean? And this is something that is difficult because ethnicity does not mean that you are from a specific country. Ethnicity is not associated with countries directly in a sense. Ethnicity is a set of activities and beliefs and daily habits and inferred relationships that you have with your surrounding community that may have nothing to do with where you are located. If you were, for instance, have strong German roots, your German ethnicity may be located in a state in the United States instead of in a place in the middle part of the Western European area. So we're probably got some things out there that eventually are going to be worked through, will become more accurate and more usable in the future. But presently, it is a very useful tool for working with immediate family relationships going back 1 or 2 or 3 generations. And in a lot of cases, you can go further back depending on the amount of other research, as I call paper research that's been done by creating the information or finding the information in documents and other sources. So what we have here is a rule that I've tried to say is that DNA testing results are particularly useful only when accompanied by a well-researched family tree. Now, this can be modified, and there's some discussion about this, but there are ways that you can generally assign people to a family line, for example. But in order to clearly and accurately delineate who a person is, you really have to communicate with that person either through a family tree that they have put onto one of the websites or by talking to the person and determining the ancestor. Some of the newer programs and newer technologies, when there's a lot of family tree information available, can begin to identify the remote ancestor. And these are some of the things that are being developed as we go along. And I like to say that without a well-researched family tree, a DNA test merely satisfies a curiosity. Now, that could be your well-researched family tree, in which case DNA may be helping you immensely to identify all of your different and verify some of your supposed relationships. But as far as a person who has no family tree and no information about their ancestry, a DNA test is not going to be very beneficial, especially when they get a long list of people that they've never heard of and have no way of determining how they're related to those people. Now, the unless here is always what I just said, and that is unless you have done the research and you have a firm structure of a family tree that's well verified, well substantiated by sources that can help you to identify and locate on the family tree where you are located to a person when you get a certain DNA match. Starting out with the history, we need to know that today, in today's world, that molecular biology and genetics have advanced university degrees, and they're advanced university degree subjects. So in other words, a person can go to a university today and get a PhD in molecular biology or genetics and a lot of other associated subject matters. And so there is an awful lot to learn about genetics and about how DNA functions, and it can get extremely technical. The question arises is how much of the detailed information is necessary to use DNA testing in a genealogical context? Well, the answer varies. Obviously, there are some people who are experts and have backgrounds in either biology or genetics or both, and they have the ability to really get into and discuss some of the finer distinctions that are happening in the DNA testing area. But the average person, when they get their DNA testing results, probably doesn't have that information or that knowledge about DNA, what it is and how it works, and what all the terminology or jargon, as it's referred to, is all about. The ultimate question is, can you penetrate the genetic jargon? Jargon is a term that's used for any specialized language that develops around a particular activity or group or interest. So if you're a photographer, for example, there's a whole bunch of things you talk about as a photographer. If you're a teacher, there's a lot of things you talk about as a teacher that no one really would know about or understand, and a lot of terms that may be very specific. And that's here in genetics and in biology and in a lot of these subjects that have, as I mentioned above, advanced university degrees that have developed very, very, very heavy jargons. As an attorney with 39 years of practice, I can testify that there is a very well-developed jargon for the law, practice of law. In fact, on the first day of law school, it's usually a mad rush from the classrooms to the library to get dictionaries to find out what the professor had to say, because even the students who are there at school cannot understand most of what the terminology that's used by a law professor. And this is very much the case with DNA. DNA is really impenetrable as far as the amount of specialized terms that are involved. Of course, we have today a lot of people out there who are talking about DNA and teaching and everything, and I'm one of them. Here I am. I'm giving a discourse on DNA, and you could well ask, well, what do you know about the subject, or do you know anything? And my answer is, well, I know quite a bit about the subject. I've been familiar with it for many, many years, but I've spent the last few years studying it in detail. Does this make me an expert? And the answer is no, and I do not claim to be an expert, but I do know history. And that's why we're talking about history today rather than about the details of DNA. Will I get to the point where I will be able to discuss that? Yeah, I'm already there, but I'm not going to claim to be an expert. And I guess the question we should ask ourselves is, how many of these genealogical DNA experts have advanced university degrees in molecular biology or genetics or any other subject that might qualify them? Well, that's kind of a legalistic way to look at things. Obviously, people who have studied and have practical knowledge in how to use DNA for genealogical research are qualified to help out the rest of us get up to the speed where we can understand and be able to use our results of our DNA tests. One of the old sayings that they said about lawyers was that to become a lawyer, you need to learn how to make a noise like a lawyer. And I guess I could say about DNA experts is to become a DNA expert, you need to learn how to make a noise like a DNA expert. I would say that's the same thing for being a lawyer. A lot of lawyers criticize because they can talk language that you possibly can't possibly understand. I had a class just this last week, and someone asked a question about probate, and within a few minutes, most of the people were raising their hands and asking questions and trying to figure out what I was talking about. So it's not unusual, and there's all sorts of areas like this. And it's one of the nice things, actually, from my perspective about genealogy, is that we do get into all these different subjects, and that genealogy continues to be a challenging and very, very difficult to understand field. Okay, so now we're going to go back into some history here. The longest family tree, at least that I was able to research, is from the present back to Confucius, back to King Tang in 1675 to 1646 BC. That's a lot of generations. I didn't get a number out of the generations, but it's a long history. Of course, if you're a relatively research genealogist and you know a little bit about family trees, you would certainly question the accuracy of a genealogy that was that long. Anyway, that's the one that's recorded, and I understand that it may be quite accurate. Both Hippocrates in 400 BC and Aristotle in 350 BC had theories about inheritance. So the idea of how people inherited their physical traits was the subject of speculation by Hippocrates and Aristotle. When we go back to Aristotle's theories about how the characteristics of people were transmitted, he was pretty correct, actually. He guessed how it was worked. He figured that there was some substance that was passed from the parent to the child in all of the different parts in all the animals and plants that determined what their characteristics were about. Well, that theory was kicked around and actually was very current, clear up until just a few hundred years ago. In fact, many of Aristotle's beliefs lasted until the 19th century. It was only the end of the 19th century when the last of the medical issues and a lot of the other things that had been first put forth by Aristotle that long ago were finally modified or abandoned. So he was a fabulously influential person. Back in 1856, and this is really when it all starts, between 1856 and 1963, a German monk named Gregor Mendel planted a bunch of peas in a garden and began hybridizing the peas to see what the results were, to try to isolate what characteristics were passed from what generation to each generation. And he came up with the idea that there were what are called dominant inheritances and recessives. So these were the idea. And all this came about, the whole structure of it. And he's called the father of modern inheritance or biology and all of and genetics. His paper that he wrote was presented at 2 meetings of the Natural History Society of Brno in Moravia on the 8th of February and the 8th of March in 1865. And they were published in 1866 and then immediately ignored and forgotten. It was not until almost 150 years later that everybody found out who Mendel was, and people began discovering his paper and realizing that he had identified the mechanism for transmitting inherited characteristics from one generation to the next. So he was he is recognized as a very important person. Unfortunately, during his life, he was completely ignored. So it was not, he never benefited from that, from having discovered all of that information. By the way, I have a picture in the background that I showed a second ago. And I just want to remind everybody that almost everything you think you know and everything you may have seen depicted in movies, books, or other media about caveman is totally wrong. Most of the things you talk about as being primitive are not that primitive. In fact, many of us who have spent a lot of time outdoors probably would not be that uncomfortable living around cave people. If there ever were cave people, I know they did live in caves, but that was just one place they lived. It's just a side note there. Couple of other theories that came down. One that was passed down by a number of writers was called the homunculus. The homunculus is that there was a theory that inherited characteristics were actually inherent in the individuals, that every individual had inside of them little tiny, almost invisible people who were fully formed and looked just like little people in there. And that when they were born, one of those little people got born, and that was what passed on the genetic material from the parents. So, when microscopes started to come in and people began to do some research and look a little bit, they could never find these little people. And pretty soon the homunculus idea kind of bit the dust. And then we had Charles Darwin, of course, who came along. And Darwin had his own theory. He came up with a theory of pangenesis, that there was some substance that came from different parts of the bodies that determined your characteristics. And part of the problem at this point in time, and this is in the 1800s, the mid 1800s, was the issue of whether or not it's called nurture versus nature or nature versus nurture. And that is that whether or not inheritance was something that came from generation to generation and or was modified by external circumstances. And of course, Darwin became famous by writing a book called Origin of the Species, and in which he raised the theory that inheritance was the changes in species or changes in the appearance of the various animals and people and everything else was due to environmental impact on the individuals. So there was always, and there are still, by the way, an ongoing argument between whether or not certain traits, characteristics are inherited or whether they are acquired because of the surrounding environment in which the individual or entity finds itself when it gets born. Interestingly enough, chromosomes, the word chromosomes, and the actual little physical chromosomes inside the cells weren't even discovered. Nobody had ever seen them or even knew they existed until 1882. So connecting whatever was happening with human cells was entirely and completely dependent on the technology of developing microscopes and other types of viewing machines that could allow people to see that there was anything down there. In other words, the reason why these people back in the mid-1800s did not know about the contents of cells is because they had microscopes could just barely see the cells, much less what was inside the cells themselves. So it was hard to discover. But the word chromosome and the idea of its existence and the little things inside of cells was Walter Fleming, who found that in 1882. And you're finding that you're going to realize here we're jumping ahead rather quickly because these developments of understanding what was happening with cell division and with inheritance from the parents to the child were slow at first to develop and then picked up speed. And now the developments are coming overwhelmingly rapidly. But back then it took a while to even get the idea of chromosomes as part of the whole issue of genetics and inheritance. In fact, there was a big backlash, and some people basically denied they had anything to do with genetics for a long time. It wasn't until 1900 that Mendel's work was rediscovered by three different botanists, kind of simultaneously, Hugo de Vries and Karl Correns and Erich von Terschmark. And these people have found it and became very much advocates, telling the world that, oh, Mendel had done this back in the 1860s, and we're just now discovering it. And so it's kind of interesting, but it still wasn't accepted, by the way. There was a tremendous resistance by the scientific community to the concepts. It was reading the history of, and it's very interesting that you might want to read the history of genetics, but it's interesting to see how much resistance there was to any of these ideas. Every time somebody said, oh, there's chromosomes, or somebody said, oh, Mendel was the guy who invented all this, there was always a major amount of resistance out there in the world. And about this time, in the late 1880s, and clear up until the 1940s, there was a movement called Eugenics, and it was not based on any kind of scientific reality. It was based on some theories that were developed by some people over in England and in Europe, and then transported over to the United States. And this is really the dark period of inheritance genetics and anything having to do with this problem. The DNA does not really come up in this until well into the 1900s, but it was well into the 1900s when DNA was being considered to be the transmitting agent of inheritance. And it's probably important at this point to mention that DNA is a chemical. It's short for, and I'm not going to try to say the whole thing out of my head. I'm sorry, I'm not doing that. But whatever, but it is, it's actually it's an acronym. So, but this is the way we refer to it as DNA. And this particular eugenics was an attempt by some individuals and a fairly large number of individuals who believed that the human race could be improved by selecting which people were able to have children and which people could not have children. Ultimately, it resulted in the people who believed in this process in doing things like killing off the people that they didn't like. And that was what resulted in the Nazis and the Holocaust in this whole area. And all I can say about reading about the eugenics movement is that it is really horrifying. It is the most terrible thing you can imagine. And it's everything about race and everything about any of these particular related kinds of things are outgrowths of this eugenics movement that began in the 1880s. A lot of the problems that exist. They existed before in a lot of ways, but here's where it became associated with sort of a pseudo-scientific backing, and people carried it to a totally horrible extreme. And it's important to understand that this DNA, the issues with DNA have this background. In other words, this is not something that's benign. It's not an issue. There are many people who will use the holdings, the findings of the genetics to attempt to establish their own agenda for racial purity or whatever else they want to call it. So now we're going to skip ahead. And 1902, Archibald Garrod observed that the disease alkatonuria was inherited. In other words, he just demonstrated that this particular strange disease, that by the way turns your urine black, was inherited from, it was inherited. And he proved it according to the rules set forth by Mendel. And this was a big breakthrough because this was the first time that anybody actually thought that a disease was inherited. People thought maybe it could be, and it was kind of obvious that certain traits ran in family lines, but there was no question when he came to this conclusion that they had finally decided that this was possible. Now, understanding that how this occurred, the mechanism for transmittal of the inherited traits was totally unknown. Nobody knew how this information was transmitted from a parent to a child. It just wasn't there. So then it slowly changed. I mean, things started changing really quickly after in the 1900s. And a Danish scientist named Wilhelm Ludovic Johansen, who lived from 1857 to 1927, coined the word gene. And they kicked that around. It was as to what they wanted to call it. And they had a lot of different variations in what they do, but they were able to, the idea here is they didn't know what the gene was, but they knew that this is the causative factor, the transmitting factor of the inherited traits. And so they called it genes. And so that's where we get our, it's in their genes, or, all that kind of stuff comes from, that began about in 1905. And that was still, as I mentioned, previously was not well accepted. Nobody really grabbed hold of that idea quickly and said, oh yeah, this is wonderful, we love it. And they argued about the name, and they argued about whether it was correct. And there were still a lot of people, scientists out there who simply said there's got to be some other characteristics. They had, usually there was a lot of argument about in a vacuum where nobody really knew anything, but they then began the process. So in 1910, and if you were to put parallel along, if you were right to parallel the history of the increase in the power and magnification and resolution of microscopes and put it alongside of the developments in biology, you would see that they just were lockstep. In other words, every time there was an increase in the ability of microscopes to get a greater resolution there, and from photography to resolve those things, the ideas began to focus more on the inside of what happened inside the cells. And they, in Thomas Hunt Morgan in 1910, showed that genes reside on specific chromosomes. So they knew the chromosomes with the little wiggly bars inside the cells, but they didn't know that there was, that the idea of the genes was that these genes were there in the chromosomes. Now, they didn't know how they were there or what they were, but the idea was that there's something in there that was inside the cell. I don't think I put a slide up on this, but a physicist by the name of Schrödinger wrote a piece, a speculative piece called "On the Origins of Life," and he expressed the opinion that that inherited traits were transmitted by chemicals, by some kind of chemical composition. That was best guess of anybody'd had at that time, and his paper was immensely influential eventually in getting people to look down into these little tiny structures down there. What the big problem that they had to overcome here was how all this information was transmitted through those little tiny squiggly things inside cells. In other words, there was no concept at the time. We today would think, oh yeah, well, it's just like a computer. It has like bits and bytes, and you could construct information and send information. But those concepts did not exist as such, and particularly were not accepted in the biological and genetic community. So we jump another 10 years or so, and we get to Frederick Griffith. He showed that genes could be transferred. In other words, that there was an actual substance inside the cell that could be transferred by a parent to a child. And in this case, the transfer of the stuff inside the cell, in the chromosomes and the genes, was in bacteria primarily. They were easier to work with because they had very comparatively simple internal structures, and they could see that this was the substance that was being actually transferred from one cell to another. And eventually, what they did was they were able to isolate the chromosomes and isolate the substances that were in them, and then by transferring that substance to other cells, they could see what the effect was. So there was, it became to be extremely complicated. Now I can actually use the term because I can look at it and make sure I say it correctly. It's deoxyribonic nucleic, nope, can't say it, sorry. Yeah, deoxyribonucleic acid, DNA, and it's probably mispronounced anyway. That's the problem with what I'm doing, is that I do this all out of books, and I should be watching more videos, because then I'd hear the words more. It's not unusual in my life that I've learned a lot of words that I've never heard anybody say. In 1944, DNA was identified as the substance responsible for the genes. This was if there's a long history of this and a lot of back and forth and real lot of bad feelings and a lot of fights and infighting and everything else. This was not something that was just, oh yeah, that's right, and everybody agreed. There were people who were absolutely adamantly against any of these ideas. But anyway, there were, after they fought their way through this process, the problem is that this is a rather simple compound. It is not a very complex compound, and it is. And it was very, very difficult for people to believe that this very simple compound was the one that was the basis for all this really complex transmission of information. Today, we probably wouldn't have that much trouble because we can see ones and zeros do a fairly good job of sending out a webinar and my voice and everything else that goes with it. But back then, it was very, very difficult to think that a very simple structure of only 4 elements was really what was at the basis of all of the transformation and the transmittal of all this information. This was done by Oswald Avery and Macklin McCarthy and Colin MacLeod, and these 3 were working and a little bit independently, but mostly through Avery, came across and discovered that this was really the basis. And it was not something that was universally accepted, but it was very persuasive and eventually became well accepted. And you look at the date, you'll see that the date was in the middle of World War II. And a lot of this was going on while the world was kind of collapsing in World War II, and a lot of terrible things were happening. And it was only this that they began to see that how the structures actually worked. So now what happened, that set the stage in 1944 for some advances that started to occur after the war ended, and people began to be more aware of the possibility that the information was being transmitted in some way. And so they were trying to figure out how this molecule of DNA would be able to be structured so that it could transmit the information. So over a period of a few years, a laboratory that were primarily in England were developing more and more sophisticated methods of photographing small crystals. The idea here is that if you're trying to take a photograph of a living cell, we have a little bit of a problem because they are always moving, and taking a picture of the cell with any accuracy was extremely difficult, and particularly. So they began developing more and more sophisticated photographic equipment, and they began to use what's called X-ray photography. The X-rays being high energy rays that had a very, very, very short wavelength, so that they were able to resolve the structure of very, very small objects. And eventually, people who were involved sort of tangentially were Francis Crick and James D. Watson. And this is really an interesting story. And if I were going to recommend anybody read about the history of the discovery of DNA, they should at least read about Crick and Watson. They were kind of, they were young people when they did this. They were in their early 20s, actually. And they were not necessarily associated in any way with any, they were associated with major research, but they weren't recognized academic people, and they were, and Watson was not, he did not even have a real background in biology. So in fact, if I recall correctly, his degree was in ornithology. So there's really not, these people were simply self-taught primarily. And their idea was that there was a way to model how this molecule worked. And fortunately, they came into, I'm not that far yet, they came into contact with a photographer, a lady who was working on this project, who is working with X-ray photography and with crystals, and particularly with DNA, and had in a crystallized form or dried form. And she had taken a series of photographs which were shown to Watson, and he recognized the structure from the photograph, and he and Crick went back and created what is now known as the double helix. And that double helix was the basis for their getting Nobel Prizes. And Franklin, who was working on this, not only was she a woman, but she managed to get cancer and die before the Nobel Prizes were awarded. And the Nobel Prizes are only awarded to living people, and so they wouldn't give Franklin a Nobel Prize, which of course I think is bad because she's the one that actually discovered the structure. But if you listen to the story, it's not quite as that straightforward, because Crick and Watson, she wasn't really looking to build the structure. She was not modeling the structure. And Crick and Watson were out there trying to get a three-dimensional model of the structure. And they were the ones who were successful in taking the information and turning it into a model that worked. And so what we have today is a model of how DNA is structured, and we come up with what's called the Human Genome Project. Now, the genome is the general term for the entire set of instructions that make up humans. And in the Human Genome Project, the first sequences were published in the nearly complete draft from February 2001. Now, here again, we have a situation where the advances in this particular area were totally dependent on the increased advances in computer capabilities. So originally it was a microscopes and seeing the structures, and then you could see it, but then nobody could figure out what it was made out of. And so then they got more and more sophisticated imaging devices up to X-ray photography, and that X-ray photography then gave them the view of the structure. And then they had people who were able to jump from that to what it is, what's it's made out of. Okay, so they came up with what it's made out of and how it was structured. And then they said, well, now let's decode it. So where are all these instructions, and what do all the instructions apply to? And so they said, well, we're going to take one sequence of human genome, and we'll do it. And we're talking about billions and billions of instructions. The amount of information in the DNA is just absolutely overwhelming. And to go through and actually count this would have taken lifetimes. But of course, by 2001, there were some sophisticated computers, and using sophisticated computers and programs, they were able to sequence and tell what the set of instructions were for that one human genome. And then the sequence was published back in only 2004. Now, I mean, we're kind of get right here, folks. This is before this, using DNA for a sophisticated purpose like trying to match who was your ancestors would have been very, very difficult, if not impossible. Okay, now we're going to jump back in time, because there's a whole nother part of this, and I'm going to have to move pretty quickly here. There's a whole nother part of how this came to be. We talked about the science and the scientists and guys. That's sort of a quick overview of it. The real issue here was, how does this become accepted? How does anybody believe that this information that is contained in the DNA would in fact actually identify someone. In other words, the first threshold here was how can you tell the DNA, why is DNA unique? What if it's the same and everybody has the same DNA and there's some other process that makes us all look different or act differently or whatever? Well, we go back now to the question of fingerprints. So we go from fingerprints to DNA, and now this is in the legal and social and cultural acceptance of this process, not just the scientific theories and all the stuff that had been discovered by the scientists, but now the question is, who's going to believe this, and who would understand it enough that they would ever use it for any purpose besides saying, oh yeah, you're a scientist, so you know all that stuff, so what, kind of thing. So we have now the question of fingerprints. Well, fingerprints have some very interesting characteristics. First of all, they had to be established before they were actually used in criminal cases and in, by the way, that fingerprint card that you're seeing in the background, it was Al Capone, the criminal. So that's his fingerprints there. But they had to have been accepted as unique, and they had to be accepted as being able to be stored. In other words, not only you say, okay, yeah, everybody has unique fingerprints, but how do we know how to use that for identifying somebody unless we have a huge bank of stored information about people's fingerprints, and we can match them up to who they are. So this has kind of gone forward. So like when I was in the Army and I was in military intelligence, so I was in a secure area, they used to fingerprint me about every month or so. I don't know if they thought I was going to turn into a different person or not, but I think they just did that to keep up on their fingerprinting. But I had to be identified every month and go up and squish my hands through the black stuff and put it on the card. So I've been fingerprinted, I don't know how many times, at least probably 30 or 40, maybe more times. But what happened is that the first modern use of fingerprints for forensics, the term used for scientific involvement with criminal investigations, was suggested in about 1863. A French professor wrote a treatise. His name was Paul-Jean Collier, and he said he thought that fingerprints were unique and that they could be used for identification purposes. Well, a little while later, like 30 years later, an Argentine inspector by the name of Eduardo Alvarez actually used fingerprints to identify a lady who had murdered her two sons and denied she was responsible for the deaths. The case is out there. You can read about it. He took a bloody fingerprint off of a door and matched it to her fingerprint, and it was instrumental in getting her convicted, even though there was no other real evidence to prove that she was the one who did it. So that was, and that case down there was not in isolation. There's nothing like this happens in law. Nothing happens within isolation. If somebody out there decides a case, then everybody in the world's going to know about it because it's a way to do things. And in the first criminal trial in the United States, took place quite a bit later. That was in 1911, People versus Jennings. And it was the first case in the Northeast district of the district courts where a criminal case went and they ruled that fingerprints were actually could actually be used as evidence in a criminal trial and upheld an appeal on the question of uniqueness. There was always the argument that how do you know there isn't somebody else with the same fingerprints out there? And so they had to come up with some enough proof and enough of a database of these, if you want to call it that, of fingerprints, a fingerprint file, to get to a very, very high level of proof that everybody had unique fingerprints. Well, you can begin to see here why this is related to DNA and what happened with DNA, because the same thing happened with DNA. When DNA was first used for identification of a criminal, there was a lot of backlash against it. I threw this in a little late, but here it is. Her name was Rosalind Franklin, and she was the one that actually discovered it. And it was because of that they were able to go forward with the DNA. Here's the code. This is the DNA code. It only has 4 components, and each one is a chemical that goes into or molecule that goes into the making of DNA. And so the question comes up, how much of the technical, detailed, scientific side of DNA testing do we need to know genealogical research? So how much of this do you have to learn? Do you need to know all these little letters and numbers and how everything matches up and all that? I'm going to have to say that actually the more you know, the better it is. In other words, it's just like anything. If you only know a little bit, it might be helpful, but if you know a whole lot, it could be quite a different deal changer. And that's the way with DNA. The more people know about it, and the more they know how it works and why it works, it becomes more and more useful. So today we have a rather large number of companies doing DNA testing, and a smaller set of those who are directed at the genealogical market. So when fingerprints came out and were unique and could be used for identification, then it was much easier to go into court and claim that here we have a different thing, only we're calling it DNA, and that we can recover this DNA from very small portions of substances left by a person on the scene of the crime, hair or skin or blood or anything else that's left there contains this DNA. And that if it's tested properly, and we go through the whole legal procedure of how you preserve evidence and make it available at a criminal trial, then fingerprints led to court cases being decided that fingerprints were possibly being used. And then DNA was then accepted as a valid way of identifying people. That had to happen before the genealogical companies could decide to sell their products and help us to identify relatives. If we didn't believe that the DNA worked to identify relatives, it'd be pretty hard to sell DNA tests. So that was something that had to be established, and it really happened in the courts, in the criminal courts. And the great acceptance of it today, by the way, is probably because of these crime investigation series like CSI and NCIS and all these shows on TV where they use DNA regularly to solve totally, in fact, they're very unrealistic, but they've managed to make DNA a big deal. And so that's how it is. Oh, by the way, here's a side thing. Identical twins are not really identical. They don't have identical DNA. For years, everybody said that identical twins inherited the same DNA because they came from the same egg and were divided up, and how could they get anything different? But later tests today have shown that they're actually not. They don't have completely the same DNA. There are differences. They're just not very much. It's slight, but there are significant differences. Okay, so genealogical DNA testing is also coincides with paternity testing, which first started with a blood test. So they didn't know anything about DNA. They didn't know how to use it. There was no really available testing procedures for it. And so back in the 1960s, they began to test the blood types. And because of very distinct blood types and some of the contents of the chemicals that were in the blood, they were able to come up with very highly accurate paternity testing. So that kind of broke the barrier to using this for establishing family relationships. And then later, as DNA became more and more into use and confidence level increased, then DNA replaced the blood tests that were used for paternity testing, and now it's all done with DNA testing. So let me ask this question. Do you have more or less confidence in DNA testing than you have for weather forecasting? Here in Utah Valley, up here against the mountains, the weather forecasts are running a pretty low level of accuracy. In the last couple of weeks, they've predicted sun and no rain, and had it rain and get rain and no rain and a lot of sun. So they're just sort of not able to tell. But it's interesting that they both work on probability. Everything that we're talking about today in DNA is based on probability. They're estimates, they're percentages, they're meaning that you have that possibility of a relationship. They're not absolute, this is 100% complete products. Here we are. The first company to provide direct-to-consumer genetic testing was GeneTree, which is now gone, and it didn't offer multi-generational genealogy tests. It was just a genealogy testing company, but it sold out to genealogists. And GeneTree sold its assets to a Salt Lake City-based company called Sorensen Molecular Genealogy Foundation, which originated back in 1999. And they did sell tests, and they were fairly expensive. And finally, another company was founded to do the same thing, named Family Tree DNA in 2000, and founded by Bennett Greenspan and Max Blankfield. And they were the first company dedicated to direct-to-consumer testing for genealogy research. They're still doing it. It's still out there. Family Tree DNA is still in business. In 2007, 23andMe was the first company to offer a saliva-based direct-to-consumer genetic testing. So it's only been, even less now, just going on about, it's going to be a while before it's even into its fifth year of having actual tests. So from going from 0 to about 100 miles an hour in about a half a second, the genealogical testing of DNA has gone from just maybe one or two small companies up into a major industry and sold all over the world internationally. So what's helpful? A well-researched family tree will be a lot more helpful to you for doing a DNA test than if you just simply want to know what your ethnicity is because you have an interest. And that goes into a DNA test plus online comparisons, because these companies can give you suggested relatives and an estimated relationship, which then begins your process of analyzing that information, contacting people, and establishing another venue for verifying relationships between individuals. And then that DNA test then can produce useful results. So DNA does, as I mentioned right at the beginning, is not overly helpful in the abstract, in a vacuum, but when put together with good research, well-founded in documents, DNA can become an extremely valuable way to establish relationships between individuals. I am discussing this online in my blog, Genealogy Star, and it is an ongoing series, and I've already fielded a lot of comments back and forth on that, and you may find out that at some point you may decide I do or do not know what I'm talking about, but this is an exploration, and it will go on indefinitely. I've been writing about it for some time, and here's the link to my blog. It's on Google's Blogspot, and thank you for watching today.
It doesn't look like we have any questions now, but the chat box is open. You're welcome to type your questions in. Our next webinar will be with Bob Taylor. That'll be on the 15th at 3 p.m. Mountain Daylight Time. That's a Thursday. If you have any comments or questions, you're welcome to email at fhl_webinars@byu.edu or visit our website, which is fh.lib.byu.edu. Thank you so much, and I hope that you have a wonderful weekend.